Hello everyone, it's Chow here, and today we are going to talk a little bit about fertilization and development, so let's get started. Okay, so developmental biology is nothing really more than the study of the processes by which animals, or also plants, and various organisms ultimately grow and develop. So in the earliest stages, we often have this term called the embryo, which is this unborn or unhatched offspring that's found within the pathway of development. So development often is broken down into sort of four different processes. We have, first of all, determination, and then differentiation, and then morphogenesis, and then we also have just growth. So if you look at this wonderful GIF over here, you can see these processes slowly playing out in motion. And I have the link over here, and it should also be found in the description, so you can go to the website itself and take a look there for yourself. Okay, so let's take a look a little bit at these specific terms themselves. So first of all, determination, which actually means there's a setting of the fate of the cell. And you can see that determination actually begins really far back in many instances, way back over here around the formation of probably the Morula stage, and it goes all the way into the adult. Next, we have this thing called growth, and growth is kind of pretty straightforward, I think. So you can see over here, growth starts, at least according to this particular figure, around the Blastula stage, and it goes all the way into the adult, and so that, I think, is pretty straightforward. We also have this term called differentiation, where it's just a process by which the different cell types arise. So we often think about things like stem cells and how they can create different cell lines, and how that one particular stem cell can potentially give rise to many different types of tissues and all that. So that's differentiation within cells, which, as you can see, really kind of happens around the Gastrula stage. Again, these are somewhat arbitrary in many instances, because you can have differentiation that are occurring perhaps a little early on. So take it with a grain of salt, and take it how the professor perhaps wants you to know. And lastly, we have morphogenesis, and morphogenesis is just the organization and then finally the spatial distribution of those differentiated cells to make a certain body plan or body shape. So I think that's pretty straightforward. Now, if we're looking at the progression, I guess I should say, of development, we can see sort of this change in the potential of the cells. So at the very start, we have that fertilization event, and then, of course, cellular division. We start off with these totipotent stem cells, and these totipotent stem cells can actually develop into a complete embryo. Once we get past a certain stage, there are going to be pluripotent stem cells, and the pluripotent stem cells, each cell can develop into several different cell types, such as different cells within the different germ layers. Eventually, later in development, we see the formation of multipotent stem cells, and the multipotent stem cells are the ones that basically can only establish cell lines within a specific tissue. So for instance, a mesoderm cell can only produce connective tissue, something like that, but it can't necessarily produce something that's a little bit different, like it can't contribute and make the placenta. So you have these steps where, as development proceeds, certain cell lines become more and more specialized. And so this is something that potentially has great impact, not just in the developmental sense, but also in terms of biomedicine. So today's stem cell research often looks at induced pluripotent cells that are generated from adult tissues. So there we go. So this is a process over here that might be good to potentially keep in mind as you're studying for your exam. So let's go through the steps of development in general, just to keep things fresh. So obviously, if you think back to middle school or elementary school, when you learned about the birds and the bees, right? That whole annoying process that they make you do in like health classes and whatnot. Fertilization is really just your sperm and your egg coming together, and then they form a zygote. In humans, the site of fertilization is near the ovary, towards the end within the fallopian tube. And once a sperm actually fertilizes an egg, there's going to be some chemical changes that, for instance, the egg will eventually block entry of additional sperm. So that prevents polyspermy, which is, in short, really bad. There's also stimulation of ion fluxes across the membrane like calcium. You can also see changes in the pH of the egg. You can see increases in metabolism as well as protein synthesis. And then, of course, there is an initiation of cellular division. And ultimately, as I mentioned, the sperm contributes haploid DNA. So the sperm is haploid, the egg is haploid. When they come together, the resulting cell is going to be diploid. I hope that makes sense. That's like intro bio stuff, so keep that fresh in your mind. Okay, then we also have cleavage, which is just the formation of many different tiny little cells that are present within the system over here. So you have parallel axes, you have perpendicular axes, and eventually these cells make more and more of themselves. And it's this process of division, but the cells are still kind of bound together in many ways and not separating and breaking apart completely. So there's cleavage, but there's not this instance where the cells just kind of branch off and go off on their own. And the cleavage eventually forms what's known as a morula. So after cleavage, you have this process known as blastulation, or the formation of the blastula. So the blastula itself is kind of divided into two main cellular groups. You have the inner cell mass over here, which eventually can develop into the embryo. In addition, you have the outer trophoblast over here. It's a sac that forms sort of the outer cells over here, and they can secrete fluid and create the blastocele. The trophoblast eventually also gives out nutrients to the embryo, and in the very end, it actually contributes to the human placenta. So the trophoblast has a very key component within development. So now after blastulation, you have the formation of a blastula, which is this little guy over here, or this little guy, and it's also known as a blastocyst in mammals. So blastula is sort of the, I think, overarching term, and blastocyst is potentially more specific towards mammals. Okay, what else is there that we have to talk about? Okay, so after blastulation, we have the next process, which is gastrulation. Now, gastrulation is the formation of the gastrula over here. So gastrulation is a very interesting process because this is the step that creates our three germ layers. We have the endoderm, the mesoderm, and the ectoderm. The endoderm in the innermost part of the gastrula will eventually form your gut lining, your liver, and your lungs. The mesoderm, which is kind of towards the middle, hence the name mesoderm or mesoderm, will eventually form your muscles, your bones, your kidneys, blood, gonads, as well as other connective tissues. And then lastly, we have the ectoderm. The ectoderm is the outermost layer, and I guess if you can think about it, it creates your skin, but also there's a part of the ectoderm that actually eventually develops into nervous tissue. So it's very important because the ectoderm can create your skin. It also affects your nervous system as well. So these are the three germ layers, and I think you should definitely know what types of tissues these germ layers eventually develop into. That's something fairly critical. Okay, so if we look at the process of gastrulation, it's kind of complex, so take this with a grain of salt just to make sure whether professors want you actually to know the individual specific steps. So here are some of the different parts of gastrulation that you can potentially keep an eye on just in case, and go look through it, read through it, and if it needs to be learned, then go ahead and learn it. If not, then you can just forget about this thing over here. So here's another summary slide of everything that I mentioned. And last but not least, I just want to say that this particular step over here, gastrulation, is fairly important. And this is definitely a topic that will come up again, not just within intro bio, but it will come up within many anatomy, potentially physiology courses, and it'll also come up on certain exams like the MCAT. So if you're planning to take certain exams like the MCAT and whatnot, these concepts will potentially come up again. So keeping that in mind, and I think that's basically all I really want to say for development. I want to keep it nice and short. If you need to know it in a little bit more detail, then your professor will let you know. And please, as always, keep an eye on your notes, keep an eye on what they need you to know, read the textbook, and as always, best of luck studying and best of luck on the exam. So I'll see you all in the next video. Thanks, everyone.